Что ты, Марго?
Не хотите? Это на вот той сборке, которая 146.
Да. Я заставлю Дашу тоже сделать бандаж. Да. Потому что даже если Леша сам все сделает, пусть потренируется, потом ей во всех остальных местах понадобится. А в бандаже глаз внутрь встроенный. Ты можешь в бандаже загнать последовательность, и он тебе найдет эту последовательность. А? Да.
Всем привет, слышно ли меня?
Да, слышно.
Отлично. Так, сейчас минуточку я настрою тоже, чтобы я видел чат. Так, все. Значит, у нас 6 человек. Давайте еще подождем. А сегодняшнюю лекцию на русском или на английском будем вести?
Скорее всего, на английском, потому что, да, у нас есть пара студентов, которые англоговорящие.
Хорошо, да.
Но
Я...
если вдруг они не придут, можно, наверное, и на русском. Хотя...
Да, у меня, которая на русском презентация, думаю, она у меня более свежая, более полная. На английском у меня та, которая еще финном читал, это ей, наверное, года 2 уже. Ну, она, по идее, тоже нормальная должна быть. Просто если буду по-английски читать, тогда и презентацию лучше английскую тоже.
Ну да, по-английски.
Угу. Хорошо. Ну, давайте пока всех подождем, я пойду себе воды налью. So guys, if you have any questions now, let's discuss something, some organizational questions maybe. Before we started the lecture, I think we can wait for 5 more minutes and we'll start. What do you think? Should I talk a little bit slowly, or it's fine with the tempo of my speech?
It's fine.
Yes, it's fine.
It's good. Always
Okay.
good.
Because sometimes I tend to speak too fast, and maybe sometimes I just skip some important topic. Because, you know, people...
I think we can ask you.
Yes, please just stop me anytime and write me. And I've lost the chat again, so that I can see that somebody is writing. Here I can see the... Oh, we've got already 14 people, so it's fine. Let me see chat. Yes, it's here. So now I've got it on my screen as well. Maybe I'll need this one. And yes, so this will be more convenient. So do you think we can start already, because we've got 14 people, and or we are waiting for someone else?
I think we can start, and in case they just
Yes.
join us.
Yes, thanks, Anna. Yes, I think we can start very slowly. Do you have this noise? Do you hear this noise? Yes, now it's fine. It's because both of us were talking. That's why. So now it's fine. Okay. So today our topic is genomics of sex determination, but I would like to start this topic kind of more broadly, because we should think, why do we have something which we call sexual reproduction? Why did eukaryote species have evolved sexual reproduction? Why it's necessary, if any species can survive without it, and what are the forms and modes, and most important for us, what are genomic consequences of sexual of the regions responsible for sex determination and sex specific differentiation. So that's why I called my lecture so broadly. And oh my God, so now how I can, oh yes, like this. So generally, sexual reproduction was always a very vividly discussed topic in different cultures, and since people started to record their thoughts, they were discussing some ideas which had something to do with sexual differences and sexual reproduction. Even when we consider Plato from the 4th century before Christ, in his dialogues, the symposium, he anticipated, for example, he makes several hypotheses. Why do people have different sexes? And he suggested that generally, very early, people had, there were some kind of people with which had traits of both sexes, and later they were divided into two. In such a way, we can assume that probably at this time already people were thinking about kind of hermaphroditism, which was behind the original state before there were two different sexes. And in some way we'll see that he was right. And generally, very many different biologists have been thinking about this topic, including Charles Darwin and many other famous people. And because it was one of the central topics in the evolutionary theory, because it was so difficult to deduce why we have two sexes and why it's so important, because somehow many species are evolving sexual reproduction, and it seems like something really essential. Let's start with the origin of this process. As we know, eukaryotic species have appeared around 1.5 billion years ago, around this time. And maybe at this time, their origin was also accompanied by the origin of sexual reproduction, because nowadays we don't know any eukaryotes that don't have any. So we do have some eukaryote species that don't have sexual reproduction, but when we do genomics on this species, we will see that there are some sex-related genes. So it means that they did have sexual reproduction, but then they have lost it for some or other reasons. And many species can reproduce asexually, but only occasionally they might use sexual reproduction, which means they still need it. And if it's so essential, why do prokaryotes survive without sexual reproduction? The answer is they also have something similar, and something what prokaryotic organisms have, and this is some sort of pre-sex or something which was presented in life before real sexual reproduction appeared. It was horizontal transfer. Let's discuss these three kinds of horizontal transfer we can observe in nowadays prokaryotic organisms. So the first one is this F plasmid and pilus special organ, and this plasmid may be transferred from one organism to another, so it may be integrated into DNA, and it can transfer the whole DNA, the whole strand of DNA into the new organism. Another types of horizontal transfer, except for this conjugation with pilus, they also include viral transduction. Viral transduction also, it's a way how to transfer DNA from one cell to another. So in this respect, we can see that somehow sexual reproduction must include fusion of different genomes which were not in the same cell. So these were two genomes in different cells, and then somehow they are combined together and they have to live together. So first is conjugation, and the whole genomic DNA may be transferred from one bacteria to another. The second one is viral transduction, when the virus may absorb some bacterial DNA, integrate it into its genome, and also transfer from one cell to another. In such a way, they may transfer genes and do this horizontal transfer instead of just vertical transfer from maternal cell to daughter cell. In this case, we can see transfer of DNA from one organism to another. And in prokaryotes, it's a very common process. And actually transformation, what we use in the laboratory. When we induce stress in bacteria, we disrupt the cell wall, and the bacteria in these shocking conditions and tough conditions, it starts to absorb DNA, integrate it into its own genome, and use new genes. This is very important. This is essential. It's a way how to combine DNA from different cells. And this is not real sexual reproduction, but this is very important for prokaryotes. All of them have that. Of course, most of organisms, most of bacterial organisms, they live in colonies. And what they absorb genomic DNA of their close relatives. And in such a way, they kind of improve their own genome, which was damaged by different reasons. And there is a theory that sexual reproduction has appeared as a protection against DNA mutations. Because let's remember how eukaryogenesis took place. During this time, it was this rapid development of cyanobacteria, which led to oxygen catastrophe. So suddenly our planet, which was the atmosphere was reducing, and then situation changed because oxygen was accumulating and it became poisonous to many anaerobic organisms that inhabited the planet. And that's why they started to develop different mechanisms how to protect from this terrible oxygen, terrible poison, which we call oxygen. And one of the mechanisms how to survive in this condition was developing sexual reproduction. This theory was published back in 2011 by Bernstein and co-authors, and he suggested that sexual reproduction has evolved from prokaryotic transformation. Because horizontal gene transfer dominates in prokaryotes evolution, and as we know that they also have homologous recombination when we can take parts of the genome and integrate them into the host genome. When we compare the prokaryotic recombination proteins, that we see that we can find some homology to eukaryotic meiosis proteins. And the second idea is that in both cases, both in prokaryotic transformation and in sexual reproduction, organisms need stress induction. Actually, this kind of stress, which may damage your DNA, what you can do with your DNA now, you can absorb it from other organisms, but in case of eukaryote species, you may just combine it with somebody's other genome and try to repair your own DNA against someone else's DNA. The third idea that the meiosis might have been an adaptation to this increased level of oxygen, which was produced by cyanobacteria. So what are the consequences of this high oxygen content? The anaerobic organisms could only survive in refugia because oxygen can be spread to many different ecosystems, and it's poisonous only in some places which remained reducing. These organisms could survive without problem. Then because of high oxygen level, it was reduction of the greenhouse effect because the atmosphere was not rich in CO2 and C42 and other greenhouse gases anymore. And that's why it was generally this cooling of the planet, you know, this hypothesis of snowball Earth, which occurred because of oxygenation of the atmosphere and general cooling of the whole planet. And because of oxygen, it was a high amount of ozone. The ozone layer was formed, which protected from the UV radiation, and thus it allowed organisms to live closer to the surface, because previously UV was too strong and it might have destroyed many organisms. But now the many more territories were free to for organisms to inhabit. And of course, as we know, oxygen has mutagenic effect on DNA. And DNA is the major carrier of genetic information, and all organisms need it is just to protect it somehow. And as we know, in the case of eukaryotic organisms, first we have symbiosis with alpha proteobacteria, and mitochondria help us because they consume oxygen, which is very poisonous. So first, mitochondria help us to consume oxygen. Second, we synthesize steroids, and when we synthesize steroids, we use oxygen. It's also kind of way how to reduce oxygen level. Another also option that the nuclear envelope was formed, and it's also in some way protection against reactive oxygen species, some sort of reduction of this penetration of oxygen into DNA, and of course sexual reproduction. When everything already happened and mutation occurred, we can all repair the DNA through sexual reproduction, because in eukaryotes horizontal transfer became really prohibited. Okay, it's here. I already told you again, this adaptation to withstand toxic oxygen. So symbiosis with alpha proteobacteria, packaging of genetic material nucleus, synthesis of steroids and cholesterol, which we don't see in prokaryotes much, and which demands huge amount of oxygen, formation of this molecule, and DNA repair by homologous recombination, which has two different consequences. First, it's a short-term effect. We can help DNA to preserve DNA in short term, but there are also long-term effects. And these long-term effects include increased genomic diversity. But if we consider, it was necessary to evolve sexual reproduction, but this process is extremely expensive. So the eukaryotic organism must have been luxury organisms to buy, let's say, this sexual reproduction, because meiosis, as we know, it takes a lot more resources than mitosis. That's why some groups of higher eukaryotes, only very small amount of, not small amount, how to say, like only high eukaryotic organisms and our model species we usually use and ourselves, we use exclusively sexual reproduction. So we excluded all kinds of asexual reproduction. But most lineages, if we will consider really basal lineages of eukaryotes, we can see that most of them use both ways. They use sexual and asexual reproduction, like protists, sponges, fungi, plants. They retain different forms. For example, they may have a vegetative reproduction, let's say in haploid phase, and then they might have sexual reproduction in other phases. And very often we can see that this sexual reproduction is induced in individuals under stressful conditions. And in many cases, we can see that after sexual reproduction, organisms form a so-called resting stage. For example, if all conditions are fine, then organisms use asexual reproduction and they reproduce asexually. But when situation is getting hard and you need to do everything to survive, first thing you should do is to do sexual reproduction and produce spore which can be kept for some time and then develop. And in most cases, except for high eukaryotes, except for vertebrates and arthropods, in most cases we can see that the main role, the main idea of sexual process is not reproduction, is not the idea to produce many different organisms, but mostly it's a totally another goal, and this goal is forming new variants and forming variety. Because, of course, if one just wants to reproduce, then asexual reproduction may be much more convenient, because you don't need to spend so much resources and you can do it easily. But when conditions are hard, you don't have any other choice, because when conditions are hard, anyone who use only asexual reproduction will have disadvantages, evolutionary disadvantages. So why this cost of sexual reproduction is so high? First, we need to develop some specialized organs second, it's a cost of finding a partner, because sexual reproduction usually demands a partner. So you need to find someone, and if the population has low density, then it really becomes a problem. Imagine plants, they cannot move. They need to do anything to find a partner, but they cannot move. They fixed on the same place, and it's really hard. Another problem is high risk of infection. Sexual reproduction seems to be can be very well used by different infections, because of course when organisms interact and they exchange material, it's very good way to jump from one organism to another. And also some other risks include high risk of becoming a predator's victim, and of course another risk is that different genders, if the different genders are formed, then they contribute in a different way to offspring development. And then there is a race of different sexes for impact into progeny development. The cost of males was very well described in the book, which I first here suggest you, by John Maynard Smith. He has written a book, very nicely written, very interesting book, The Evolution of Sex. And there he discusses a lot about the problem, which includes difference in investment. And this idea, according to this idea, sexual reproduction will be twice more expensive in comparison to asexual one, because you always need two partners, but the number of individuals may be very well produced by the same organism. And biological effect of sexual reproduction, as we know, it's first removal of accumulating harmful mutations, because it's a way how to combine harmful mutations and take them away, and also to combine good mutations together, and which increases evolution and makes it very fast. Also, it's very important for generation of new allele combinations, because these new alleles might help to adapt for new changing environmental conditions. So if we have stress, it means everything changes, the weather changes, the ecology changes, and if we will start to use different combinations, maybe we will find a combination which can survive. So this is the way. If we will just keep the same combination, maybe it will be not enough to survive. But if we will start to form many different combinations, maybe one of them will become fruitful and survive. And generally, if we consider large populations, then if the population would be very large 1.5 million years ago, that probably sexual reproduction would never appear, because in big populations, probably it would be enough organisms who could survive these harmful conditions. But for small population, sexual reproduction becomes really critical. And that's why sexual reproduction was important for small populations, and it's very important for complex and slowly developing organisms. Do you have any questions? Maybe because, you know, this part is always not controversial, but it always evokes some question about this, because it's just a theory, you know, it's not something very well established, because it's all mostly formed based on different mathematical models. And generally, all these models predict that there is a chance of appearance of sexual reproduction, but actually it maybe not necessarily so, might have been not necessarily so. Maybe we could have a planet without sexual reproduction as well. Why not? But it did appear in our evolution. That's why we discussed it. Okay, and if you have questions, please write in the chat, so I will see if I really mentioned all questions. So let's now talk about something which we call Merlet's ratchet. You probably heard already some this problem from evolution. One of the founder of genetics, Hermann Müller, he suggested when he did his studies in Drosophila that there is a lot of harmful mutations because, as we know, the organisms, any organism cannot reproduce without mistakes, and anytime mutation appears, so DNA cannot be. Let me see, somehow I cannot read. Why sexual reproduction? I cannot read it. Is it?
I accidentally sent it before I wrote the sentence. I'll finish it and send later.
Okay.
Sorry.
No problem. Don't worry. I just thought that I couldn't see the whole sentence. Okay, so Hermann Müller, or it's not actually, oh, it should be U. So he's Müller, actually, Hermann Müller. And he suggested that any organism always has mutations. So any born organism has new mutations, and most of these mutations are not adaptable, are not favorable. Most of them are neutral, but maybe some may be also harmful. And he said that after appearance, this mutation, if it's slightly harmful, then it may accumulate in populations. So this populations will start to accumulate these mutations, and that's why if we will observe asexual populations, for example, cloning bacteria and these reason mutations, then whole genomes may accumulate these mutations, and sooner or later, when the load will be big enough, they will just die and disappear. So if it will be some sort of purifying selection which will take out those mutations somehow, then sooner or later, all our genome will be mutated and disintegrated. And during sexual reproduction, it's a way how to combine the genes which were not mutated or which have beneficial mutation, and in such a way, without sexual reproduction may be very big problem of this genetic load. So the question was, let me see. So why sexual reproduction is important for slowly developing organisms? Because if the organisms are very slowly developing and the care about the progeny is very important. And in this case, it's like our strategy. Remember, it was K and R strategy. Our strategy, when we produce too much offspring and we don't care, some of them will survive and it will be good. So if some of them will be mutated, why not? And we don't care much because it's such a huge number of progeny that we don't need to care. But slowly developing organisms usually have not very many children, and that's why it's important to keep what they have already generated. That's why it's so important to have different kind of combinations when you can combine and get something which will survive, and you need to spend more time and more resources for the progeny. So this is just my idea about it, but generally it's because of, it was mathematical stimulations. So they try to calculate the cost, they try to calculate if it's like organisms which are very fast developing and reproducing, or very slowly developing and reproducing. And these models, these people calculated that if the population is big and very fast developing, that sexual reproduction may never appear. But if the population is small and the organisms are very slowly developing, then the same mathematical model works in a direction that sexual reproduction appears. And about the emergence of new combinations, it was the idea was expressed back in 19th century by August Weismann, a famous German geneticist, who first suggested that new combinations appear during due to the sexual reproduction and combination of traits from both parents, and in such a way it may increase the general combination effect of different alleles. And besides August Weismann, he also formulated the vicar of Bray hypothesis. He said that sexually reproducing population produces greater diversity, and the most adapted kid will survive. And that's why when the environment is changing, that in these changing conditions, this will be very beneficial. And he compared it to vicar from Bray, who adapted to different governors of his country, and he was moving from one religion to another, depending on the belief of the ruling monarch, because this fast adaptation helped him to survive hard conditions. And also the evidence that sexual reproduction is not just for reproduction, but for DNA repair. It also comes from the fact that in some unicellular organisms, the role of meiosis as a repair mechanism is the only adaptive function. Because, for example, if we consider yeast as ascomites, some ciliates, in these organisms, sexual reproduction mostly occurs between genetically totally identical individuals. So they don't have any advantage in forming new combinations. They are identical. Why do they need sexual reproduction? They need it because it's not for new combinations, because it's for DNA repair. So in this case, they retained this ancestral function, and why probably all this sexual reproduction first appeared. Another helpful advantage of sexual reproduction is protection against parasites and selfish genomic elements. Yesterday, we discussed some selfish genomic elements, and today we bring sexual reproduction as in another way how to struggle selfish genomic elements. And this idea was first proposed by Leigh van Wallen, and he noticed that asexual populations actually are more susceptible to infections. Remember, we told that because of sexual reproduction, two organisms must meet, and this is the way how to get infected and so on. But actually, when we try to analyze, for example, if you analyze asexual, some organisms have both sexual and asexual populations. If we analyze asexual populations, then we surprisingly see that actually there are many more different parasites, different infections, and accumulation of genetic elements. Why it happens? Because the purifying selection doesn't work so well in those organisms. So they don't have this way how to get rid of this load, genetic load. And as he brought the citation from Alice through the looking glass, when the Red Queen says, now here you see, takes all the running you can do to keep the same place. So to stay on Earth, you need to evolve very, very fast. So if we have so many advantages of sexual reproduction, why do we still have asexual species? And in some eukaryotic groups, really we can observe that sexual reproduction disappears. But this is a very rare event, and maybe it may bring the whole group to the dead end. For example, bdelloid rotifers. This group of organisms, you remember who are bdelloid rotifers, right? In Russian, they're called bdelloidные коловратки. So these are multicellular organisms, and they remain to be problem and paradox and evolutionary biology. And since in these organisms, no sexual reproduction was observed, no meiosis. And actually, without these strategies, they must die out very fast, but they do persist for tens of millions years. Why is that? And another quality of these organisms, they are very resistant to drought in any stage of their life cycle. They are really resistant to ionizing radiation. And when people first sequence their genome, they suggested that maybe there is no sexual reproduction, but there is something which is called gene conversion and horizontal transfer, something more similar to prokaryotic organisms, which they took as a replacement of this important process. And these people said that around 8% of the genome was some sort of brought by horizontal transfer. But this was really very contradictory idea, and late, because now we know why we need to assemble genomes so well, because if we don't do it in the right way, then we may form really strange hypotheses, as we can see here. So 2 years ago, or a year ago probably, a group from Moscow, they found that actually there are some evidences of sexual reproduction in bdelloid rotifers. And these people have shown that actually when we analyze the genome, we can see some traces of recombination, which means that sexual reproduction still exists. Maybe it's very, very rare. It's very hard to find. Maybe they form males only as an exception and in very extremal cases. But nevertheless, they didn't get rid of the whole process totally. And it means that we just understudied this group. And actually, we need now to replace all chapters in many school books where we say that bdelloid rotifers have no sexual reproduction, because now we know that actually it's not the case. Sexual reproduction still exists in this group. Okay, now let's talk about the different ways of sexual reproduction, and start with the most primitive variant of sexual reproduction, which is called isogamy. Isogamy is a way when both gametes are not morphologically different. So we don't have like male and female here. We don't use these names. We use something like plus and minus, because the idea of sexual reproduction in most cases that they shouldn't come from the same parent. That's why we don't need two gametes to be fused from the same parent. We prefer when they come from different parent. That's why somehow, even if they're similar, the gametes, they must nevertheless some sort of carry different recognition marks to get fused. And sometimes in these organisms, they don't have any sexes, but they do have different mating types. I don't know if we can call them genders or not. This is also controversial topic about what we should call sex or gender or mating type. Anyway, in biology, we can just say mating type. And for example, in some mushrooms, there may be up to several hundred different mating types, and they're produced in such a way that like this gamete can fuse with any other except for itself, except for the same mating type, but all other 300 it can fuse with. And this is a pretty primitive strategy, and it's still retained in many different organisms, like in Spirogyra, in chlamydia monads, and in other organisms. And in many cases, very fast, when from isogamy, anisogamy evolves. Anisogamy, when the sexes are formed and different organisms start to produce different kinds of gamete. One of gametes is usually larger, and in this case we call the organism female, and the other small gamete we usually call male. And in the case when the female gamete becomes really large, it may lose its ability to move generally, and in this case we call this type oogamy, when the egg cell actually is not moving and it's pretty big and it accumulates a huge amount of materials for developing embryo, but the male gamete is really small and it will move very fast. And interestingly, anisogamy evolves independently in different eukaryotic groups, which means that there are some evolutionary advantages, that one group is producing only big gametes and another group produces very small gametes. Of course, in this case, only two sexes can exist, because if there would be a third sex, then it would be too complicated and more difficult. And with two sexes, it's way easier. And if the oogamy emerges, why don't we have the situation when the same organism produces both eggs and sperms? Why do we have different sexes? Why do organisms differ and specialize in producing different gametes? Why don't stay hermaphrodites? Because the price of males is very high. They just produce very small gametes and they don't contribute much to the progeny. And let's consider now hermaphroditism in different lineages when the same organism produces both types of gametes. And we can see that hermaphroditism we can see in snails very often, but not only in snails and worms, which we learn in the school, but also in some vertebrates and even in fishes. Thus, in some fishes, we can observe the way of hermaphroditism, which is called sequential. In this case, and around 2% of all bony fish, as we know, bony fish, there are more than 20,000 species of bony fish. So it means 2% of them, it's like almost several hundred species. And in bony fishes, usually they start their sexual career as females because they're strong enough, they can produce big gametes, but later it's getting too hard and they just already want to relax and not produce too much energy, not spare too much energy to produce eggs, and they start to produce sperms. So this is called sequential hermaphroditism, and it is observed in pretty many fish species. But also something which is called synchronous hermaphroditism, when the same organism produces at the same time both eggs and sperms, also exists in vertebrates. And this species is Cryptolebias. It's just the only species was described from Cypridontiformes. And in this case, both types of gametes are produced. And in this case, it's very interesting how the ovary and looks like. And these different parts of gonad may have function of may have female and male function. So if you are really interested to read more about evolution of sex determination in different organisms, it's my advice to you to read this book. This book was issued several years ago. It was Leo, I know him personally, so he recommended me this book and he sent me not only this picture, but the poster about this book, and I bought it in Amazon. So if you So if you're interested, and I dream that somebody will translate this book into Russian and that it will be also available in our bookshops. So the book is called The Evolution of Sex Determination. It's written by Leo Boykboom. It's a scientist from Netherlands, and Nicolas Perrin. Nicolas Perrin, he's from Switzerland. He works on, Leo works now mostly on insects and flies, and he studies sex determination in these groups, while Nicolas Perrin, he was, I think now he's retired, but he's still active scientifically, and he's working on frogs and salamanders. He has many articles about this. So what they did, they accumulated data on different groups of organisms from the whole tree of life, and they described different types of sex determination, including ploidy, in which phase sexual reproduction occurs, also the types, if it's either genotypic or epigenetic sex determination, because we know that, and sometimes that the external environment affects the individual development, and in this case it will depend on temperature. I will tell you later about it. Now we just briefly mention this. And other ways of sexual reproduction. So we, for example, we can find here ourselves. This is Theria. So these are mammals, and we can see that first it's unisexual. So it means usually one organism has one sex, either male or female. So it's unisexual. Second, it's genotypic. It means environment doesn't influence much our sex determination. So it's environmental, and it's round, which means it's diploid. So the organism which produces gametes is usually diploid. So in this way, we can analyze all different groups and see what happens there. Although some groups may be really mixed, and in some groups, really different systems exist. For example, here we can see crocodiles, and we see that crocodiles have epigenetic sex determination, which means temperature determines which kind of sex it will be, if it will be males or it will be females. Okay, so this is my recommendation. Please find this book and read it. I hope that PDF is available already, because when it first appeared, I couldn't find any PDF, so I bought the hard copy of the book. So let's see what happens in plants. In most plants, in most flowering plants, they are monoequous. What does it mean? It means that in Russian we say однодомные. It means that the same, in Russia we say двудомные for monoequous, which means that the same organism produces both male and female gametes. So usually, remember when we were discussing the morphology of the flower at school, we saw both types of, we saw the pollen and we saw the egg cell as well. So, but only 5% they are actually dioicous, which means that there is a plant which will produce only small gametes and the plant which will produce only big gametes. Why such a low number of dioicous species? It is because like in this case, for example, in Arizema japonica, it's also interesting because when the plant is very young and it's weak, it produces only male flowers. It doesn't have enough strength to produce egg cells. But when the plants are strong and large, they may produce female plants. So why plants are more likely to be monoequous? Why do they have, most of them, both kinds of gametes? In such a way they're kind of hermaphrodites? And the answer gave Charles Darwin. He said that because plants cannot move, they're stationary, they don't have any chance. For example, if you're far away from anybody and you don't have anybody to meet with, the only way you can survive, for example, you have only one seed on the island and if there is no way to find any other plants, then anyway you can use self-pollination. You can use your own sperms, your own eggs, and produce plants, and at least survive for some time using this, using self-pollination. Of course, it's not very advantageous. In the evolutionary perspective, cross-pollination is much more progressive. But for the first time, this will give you an opportunity to survive if, for example, you cannot find a partner. This is the answer. Interestingly, in plants, also some sex chromosomes were described. Very, very rare, but nevertheless, actually it's in Diercos plants happens. XY system was described in asparagus and in Selene plant, but it's rather exception than the rule. But just for you to know that even in plants, sometimes we can find sex chromosomes. Okay, now major types of sex determination. Why do some organisms decide if they will produce small gametes or large gametes? There are two main ways which determines this. First is environmental, when something from the environmental tells you now you ought to be the male, so you produce the small gametes. Or it's internal or genetic, and both of these strategies are widely used by different organisms. And of course, we will more concentrate on genetics, on genetic sex determination, because actually it's what we will mostly talk later, because it's genomics of sex determination. You know, I'm talking for such a long time. Let's make a short break. What do you think? Just to get distracted. Guys?
Yes.
Yes, we agree.
Okay. If you want, we can just talk, drink a little bit of water, because later we will talk more about genomics, and now it was like more introductory generally to think about why generally we have sexual reproduction, and later we will concentrate on genomic consequences of this.
5 minutes, right?
Yes.
You know, today in the morning I was reading lecture in the medical university here in Bishkek, and it was kind of very nice way. So it was very good and interesting conversation with students, with teachers.
Oh, that sounds very interesting.
Yes, but then of course, like it's a little bit like traditions from Soviet Union, when after this you go to the department and then you like have a conversation, and of course you drink a little bit of alcohol, and maybe not little bit, and it's early in the morning. Pretty tough.
When we were in Manas University, no one suggests to us alcohol.
Because it's not a medical. I was in the medical university, the governmental, so
they have a lot of ethanol.
Yeah, no,
Probably.
we drank vodka, but it was also strange, you know, because people are Muslims, so they pray before like meals, and then they just use vodka and like.
Interesting.
Yes, but very, very nice people, and very, very interested to collaborate, and they're mostly teaching.
I noted this too.
They also have many international students there.
From what countries?
But they have different lectures. So I had only students from Kyrgyzstan, I think, because I was holding my lecture in Russian. But I think they have different disciplines, and these disciplines are in English, and they have mostly students from India and Pakistan. And this is different from Novosibirsk, because in Novosibirsk we do have also some students from India and Pakistan, but mostly we have from Iran and Syria and Iraqi, and I don't know why such a different distribution of students between countries. But this is very good because it makes everything very international, and I hope that it will be very beneficial for both parts, participating parts.
And how do you like Bishkek in general? Like food, architecture?
I like very much. Yes, very, very tasty food, very many good places to eat out, very cheap, and also very nice people. So I think the main potential of the country, of course, it's very nice nature, but they also have very, very nice people and very high culture of communication. People are very open and very helpful, and it's very easy to find your way through the city. If you talk to people, if you openly address them, they're always happy to answer you. They're always happy to help. This is really nice. And somehow I already found my way how to approach people, and I more or less know what I will get in response. So for example, when I stay in some European countries, I know that some things are not possible. You just do this because it's a rule. Here, everything is possible. For example, if I stay in a hotel and it's not provided, no coffee is provided, you can always go downstairs and say, oh, you know, I want coffee, can we organize this? And you can find a way to organize it, even if it's not available and if it's not suggested by the hotel. But by talking to people, by interaction, you can get so many things you want.
Have you tested Kulikovsky?
Today, yes, today was, you know, in Novosibirsk, Kulikovsky is in front of my house. That's why I was visiting Kulikovsky back in Novosibirsk, and I was buying these frozen cakes and for different reasons for months. And I even have application on my phone. So when I saw here Kulikovsky today, I decided to drink coffee there. So I went there and I bought coffee and the cheesecake, and my application worked. So it's kind of application I installed in Novosibirsk, perfectly well working in Kyrgyzstan. This was really funny.
Yeah, this is really, really nice bakery, and our group now is fan of it.
But don't you have it in St. Petersburg? Because in Novosibirsk, it's in front of my house.
No,
I just go out and I buy always frozen. Like when my relatives visited me, we always bought very nice ice cream there.
Unfortunately, I have never met Kulikovsky in St. Petersburg. Maybe, but not in my area of my observation.
No. The Kulikovsky, there is no in St. Petersburg.
Oh, what a pity. Maybe you have another different bakeries. You know, in St. Petersburg, I never had problems to find a good bakery, but in those we do have this problem. That's why Kulikovsky is so popular.
I don't know why, but in Kulikovsky is very tasty coffee because it's more creamy and soft. I don't know why. Maybe because, of course, of its price.
It's because it's because of the
milk?
grapes. Yes, because of milk. Because it is milk from the mountain.
Yes, I also thought that the components are very natural. At least what I test from the cakes, it tastes very good. So it's like really natural milk or animal fat which they use. So not so much of soya or some other stuff. Okay, let's go back to sexual reproduction and genomics of sexual reproduction. So, as we already said, sexual reproduction has evolved once, And that's why this cascade of genes, which has evolved 1.5 billion years ago, it's still retained. And we can see that the same genes govern development of gonads in different organisms. In the fruit fly and in human, these genes are homologous. The mechanisms may be different, the details may be different, but usually the main genes may be homologous to each other. And in this case, almost what is also interesting for sexual reproduction, it doesn't matter, for example, human, we take male or female, mostly we have all genes necessary for both male and female cascade. It's not something that we only have, males only have male cascade on Y chromosome and female have only female. Actually, in mammals, females don't have something specific. They have always the genes that female have, male will also have, right? Because they only have additional Y chromosome. So genomically, it's something which presents in both sexes. And there are two different kind of circuits that suppress each other. For example, there is male and female cascades of genes, and in case one is prevailing and it's kind of balance, and then this balance change, we will say later, the trigger, some sort of trigger changes the balance, and then either male system wins or female system wins. And once it won, it takes over. So the winner takes it all. Which is the trigger? This is a tricky question. In our case, it's SRY gene on Y chromosome, as we will see. But in other cases, for example, in crocodile, it's temperature. So there is a balance of two suppressing each other systems, and then high temperature suppresses one of them, the second wins, and it kind of totally suppresses the opposite, and then the organism becomes female, for example. In case of sex chromosomes, in sexual sex differentiation, there is a special trigger on one of chromosome, which we call sex chromosome, and transcription of this gene leads to suppression of one of sexes, and it may be either male specific or female specific. For example, in case of mammals, so this group of genes, SRY, it interacts with SOX9, and then there is this circle of self-enforcing system. When it wins, then the male system is developing. And in case of female circle, it's totally different genes, and they might be suppressed by those genes. And in mammals, if there is no SRY gene, for example, so it doesn't matter if there is Y chromosome or not, if SRY gene is not active, the default system is always female system. So the female system will be developed if no SRY gene triggers this kind of cascade. And let's talk about sex chromosome discovery and diversity. Sex chromosomes have been discovered more than 100 years ago. First, they were discovered at the end of 19th century in wasps, when wasps with 12 and 11 chromosomes were described. And Henkin called, the scientist who found it, called the smallest chromosome X element. And he suggested that since this element is present in females, he suggested to call it X element. And since then, we call the female specific chromosome, not specific, but the prevailing in females chromosome, we call X chromosome. Later, McClure also found an additional element in spermatogenesis of grasshoppers, and he also suggested that it's connected to sex. And later, it was already ideas about chromosome reduction in meiosis and connection between chromosome theory and heredity. And later it was obvious that many different systems exist, and people started to discover different sex chromosomes because you need to have its microscope. So you take the microscope, you study meiosis in males and females, and you see the difference. So you describe the sex chromosomes. You don't need much. You don't need genetics. You don't need to do sequencing. You just need cytology to discover sex chromosomes. And talking about sex chromosomes, we must say that there is a single sex-specific element, and we call it Y in case of males and W in case of females. So this is the chromosome which is specific to only one of sexes. And usually this element is the most variable part of the genome. Usually it accumulates huge amount of repetitive elements, also because it doesn't recombine. So this Müller-Reichert works here very well, and these chromosomes start to accumulate repetitive elements. And another element which presents in both sexes, but in a different ratio. For example, X chromosomes in human, there are 2 X's, and in females and 1 X in males. So they X presents both in males and females, but in a different quantity. And Z chromosome is also similar because it presents in males and females, but in different combination, in different number. And usually this part of genome is the most conservative one. So usually the X and the Z chromosomes are much more conserved than the autosomes, so the not sex chromosomes. So how did the sex chromosome evolved? Back in 2006, it became obvious that the sex chromosome have autosomal origin. But of course, this autosomal origin has been already suggested by Müller, we already discussed, Stefan Müller, and Susumu Ono later. 50 years later, he also suggested that sex chromosomes have autosomal origin. And if we, for example, consider birds, snakes, and mammals, 3 different groups. Here they color it in red, blue, and yellow. So they were 3 different pairs of chromosomes. In 3 of these big vertebrate groups, for example, in birds, it's a very conserved system. They have ZW system, and this Z chromosome and W both evolved from the yellow group. In case of snakes, also pretty conserved, although now there are some exceptions in snakes where sex chromosomes evolved from a different pair. But nevertheless, in most snakes, sex chromosomes appeared from the only single pair, and here we color it in blue. And in mammals, sex chromosomes were formed from the red chromosomal pair. So we see that 3 different elements, they might become sex chromosomes in different groups. What happens when one of chromosomes decides that it's now sex chromosome and it triggers sex reproduction? First of all, this chromosome starts to acquire new sex determining. First, when it actually happens, when the chromosome becomes sex chromosomes, the autosome becomes sex chromosome, it acquires a sex determining gene, a trigger. So this allele becomes something which triggers this kind of balance. So it suppresses one cascade, so the other cascade wins, and then suppresses another cascade totally. After this, what happens? The recombination is getting suppressed between these elements, and chromosome-specific region start to extend. So here you see that the autosome, and they recombine with each other, X and Y. The Y chromosome acquires, in this case, male trigger allele. And then after the proto-XY chromosome, this is still autosome. This is already a sex chromosome because this is testing a testis differentiation factor, and still recombination happens except for this element. Actually, this element on this stage, it can also jump from one chromosome to another. There is still no distinction between chromosome. And then this gene becomes to accumulate some sex-specific genes, because it's evolutionary advantages. If you not only have just the trigger, but you also have some genes which, for example, are needed for testis development, not only triggering male-specific sex determination, but also, for example, genes responsible for sperm development. And then when you've got already these genes, you're not interested to have any recombination, because in this case, this gene might jump and you can, again, you can disrupt your sex trigger and this sex-specific allele. So in this case, somehow recombination suppression happens, and after the suppression of recombination, chromosome-specific regions start to evolve, and the recombination doesn't take, actually the X chromosome, it's still recombining in females, right? Like nothing happens to the X chromosome because it's recombining not so often, but nevertheless, in females, two X chromosomes meet and they recombine. The Y chromosome is suppressed, and as soon as it is suppressed, it starts to get accumulate repetitive elements, tandemly arranged repetitive elements, line elements, sign elements, transposons, huge amount of them start to accumulate, and the genes which were present on the proto-Y chromosome, they start to disappear. They start to degenerate and getting extinct. And after a while, after this degeneration, happens through the whole chromosome except for one tiny region, and usually we call this region pseudo-autosomal region. Pseudo-autosomal region is very interesting because it's the only region where the X and Y chromosome recombine, and in such a way this recombination will be really very often. Imagine if it's the autosome, recombination may happen here, 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 anywhere, but it should be one recombination per meiosis, right? In each chromosome pair, and it can happen anywhere. Here, it can only happen in a very small region. That's why this recombination is really intense. It's much more intense than on autosomes, and it makes this region very, very interesting. And the rest part is degenerating because it's not recombining, but X is not regenerating because X is recombining with the other X in females. And some people hypothesize, so Jennifer Marshall Graves is a famous scientific pessimist. He believes that finally, after all this degeneration, the Y chromosome will be disappeared because it's just stage when it hasn't disappeared yet, but it's already degenerated so much that it's almost nothing left but the trigger, the gene trigger, and some important genes. So she believes that the Y chromosome will disappear very soon, while other scientists say no, it won't happen because it can still persist for millions of years because the process has stopped and degeneration doesn't take place anymore, and that's why we cannot say that it will disappear. So people are really struggling. Several times I've been to the talks by Jenny Graves, and I saw people who were really red. They really didn't like the talk. Some of them went out of the hall because they didn't like the idea, and it's always very nice. I like, and when they started really to shout that they don't believe it, and it's really interesting because it has something to do with sex. When you have it, then you should be ready that it will be really interesting discussions about it. So what we observe now in different vertebrates, we observe different stages of sex differentiation, sex chromosome differentiation process. For example, sea turtles and alligators, they don't have any sex chromosomes. Their sex depends on temperature and recombination happens through the whole length. So it's just normal autosomes, no sex chromosomes. Please don't search for sex chromosomes in alligators and marine turtles. You will never find them. But in some other organisms, some sort of young, very young sex chromosome exists. When we know that there is a sex determination, genetic sex determination, but when we see the karyotypes, we cannot see the difference. For example, in frogs, we see the male frog and the female frog, and the chromosomes are totally the same. And in this case, the difference may be just single SNP, just one nucleotide polymorphism may be responsible for sex determination in this case, but mostly it's a gene. Mostly, of course, it's not just one SNP. But there were some cases described in some fish where it was just very small region responsible for sex determination. And in pythons, something similar, like when we observe Z and W chromosome in female python, we don't see any difference between this chromosome. We know that it's the sex chromosome, but we will find the difference only if we will sequence. I suggest, so if you deal with this kind of animal, you know, it's genetic sex determination. Usually it's very easy. How do you know that this animal has genetic sex determination? Your ideas?
Just make a karyotype and look.
But these can be homomorphic chromosomes. They will be similar. So if you have huge progeny, calculate the number. If the number is 50-50, you can be sure it's genetics. Because when you have environmental sex determination, you have a little bit hotter or colder temperature, and you never have 50-50. You understand, right? So you can change conditions and have different number of individuals of any sex.
I have a question. We are discussing that it's possible when there are a real big difference between X and Y chromosome, or
Yes.
Y and Z. But what is just the beginning of the sex evolution in this species, in this clade? And for example, in some fish, I know that it's possible, and there are Y and Y and X chromosomes, but the difference just several genes. And it's like at the previous slide, there was situation.
Yes,
how we can understand, because it is now it is a real sexual genetic variants, but we can't find it. And
yes. Thank you for your question. Exactly. This is the case. We check the chromosomes in, for example, take 50 males, 50 females, check chromosomes, they are totally the same. We don't see any difference. In this case, we say there is a case of homomorphic sex chromosomes. They're totally, the Y looks totally like the X, or the Z looks totally like the Y. How we can find those genes now? We take two pools of males and females. Let's say we take 50 males, 50 females, we sequence them, and then we compare the differences. So when we see the difference of the female group or the male group, then this difference will be persistent in different individuals. And then we can say that this is the region which is responsible for male or female sex differentiation. So you understand the idea, right? Not just one male and female. If you take just one male and female and do genome sequencing, we find very many different regions because we are all different. We all
yes,
have,
of course.
And all these SNPs, all this, they will make our task very difficult. But if we take 50 males and 50 females, we will diminish this problem. And we will definitely see some principal components, for example, which will appear only in females, but we cannot see them in males. In this case, we can say about W-specific elements. If we see them in males and don't see in females, we say about male-specific elements. And of course, we can also see the heterozygosy. For example, if we compare, even in the same individual, just one individual, we have one male, one female, we check heterozygosy across the genome. We see, for example, that this genome is heterozygous, and in one region, in male, we see that it's exactly the same SNPs from both alleles. So we can assume that probably this part is not diploid, maybe it's haploid, and it means that it's something to do with sex chromosomes. So this is another way how to search for them. Okay, So this is homomorphic sex chromosomes. The next stage is when chromosomes are still similar, but some degeneration takes place. And we can see these chromosomes in some red-eyed birds, like in ostriches, in colubrid snakes, in medaka, when the sex chromosomes are more or less similar in size, but one of them is a little bit degenerating. And this is really interesting situation. We already can find them cytologically. And the next stage we can find, for example, in all mammals, in all eutherian mammals, or in vipers, in carnitine birds, when sex-specific gonosome, of course, we can say sex chromosome, but we can use also the term gonosome. So when sex-specific gonosome is getting really small, accumulates repeats, it looks really dark when we do C-bending, so it's really easy distinguishable cytogenetically. In this case, we can say about very well differentiated sex chromosomes, and in this case, we humans are very lucky because we can easily distinguish the Y chromosome on the karyotype, and also we can easily distinguish the birds because we can find the W chromosome in female birds. And the next stage, when the pseudo-autosomal region is lost, we can also find, like in some voles, you know, this species voles, polevki in Russian, the pseudo-autosomal region may be totally lost. And the same happens in marsupials. There is no pseudo-autosomal region, but still the X and the Y, they are not recombining, but they stick together during meiosis. And this situation is also kind of intermediate situation, and the gonosome-specific sex chromosome is really easy distinguishable. And the next stage, as believes Jenny Graves, is when the sex gonosome is totally lost. And actually, we do have some examples in nature, and it's like mole voles in the species Elobius or spiny rats. And in this case, we can only find X chromosome and we cannot see Y chromosome at all. And this situation really exists in some very rare rodents. So, do you have questions about this? Okay, if there are no questions, let's go back to sex determining pathway. We already were talking about this overall picture is still not clear, and mostly all genes which are involved in sex determination, they were discovered either on mutant animals or on patients with intersex syndrome. So SRY gene was discovered in such a way. But nowadays it's easier because we can do transgenesis, for example. We can induce some transcripts, or we can introduce some genes, and we can change sex because of this. And this is the best way to prove if one gene is triggered or not. We just need to induce this gene on particular stage and see if it changes the development. So generally in mammals, it exists in such a way that B potential gonad, which is affected by different genes, its development later develops into either testis or ovary, depending on the trigger. If SRY triggers the development, then the balance will lead towards testis. If no SRY is included, on default, this cascade will win, and RSPO will interact with WNT and DAX gene, and ovary will be developed. And generally, now you're involved in so many genome sequencing projects, which is really fascinating. And for us, it's a unique opportunity to compare genomes from different parts of the phylogenetic spectrum. And we should always remember about sex determining parts, because they behave differently in evolution. And we must remember that in different taxa, genes from the cascade can be expressed at totally different times. Male and female expression levels may differ of these genes, and those sensitivity may also vary. And probably because mostly when we analyzed genes, sex triggering genes from many different animals, most of them have appeared from this trigger. So most of this and only some of genes, actually it's more than 30 genes in each part, and usually one of these genes becomes sex determinant. Like SRY gene has appeared as a paralog of SOX3 gene, but I will tell you later about it. So any of the genes, because of evolutionary instability, may form an allele which will change the balance, and in such a way, this allele may acquire the function of the trigger genes, And this happens in different lineages of vertebrates. And in vertebrates, we can see, for example, that eutherians are pretty boring. It's just XX, XY system. And in snakes, for example, also not very interesting, just ZZW, but they also recently, it was one species discovered with XY. Lizards are much more interesting because we have all possible variations. We have temperature-dependent sex determination, we have ZW, we have XY, and we have mixture of all of them. So it's really interesting group. So if you are working with the genetics, with the genomics of this group, it's really interesting to see what kind of sex determination is there. And in most cases, it's not clear. In Anolis carolinensis as XY system, and it's very well described. In other lizards, it's not so clear. In birds also pretty stable, ZZZW. For some reason, in some groups, the system really appears once and they are kind of, they're inside the trap, evolutionary trap. They cannot move. For example, mammals and birds, as soon as they acquired this system, they cannot evolve anymore. But other systems like lizards, it seems that they can change this strategy very fast. The same with turtles, with amphibians, with fish. It seems that these groups are much more plastic. They can adapt. Their systems are more variable, adaptable. They can change sometimes within the same species from one system to another very easily. They are not trapped yet. Rather than mammals and birds, we already cannot do anything with this system, because as soon as we start to do experiments, it ends very bad. Because the problem with sex determination, it's a cascade. It should be very precise, either male or female. If we have any intermediate option, it's not adaptable. It's not advantageous. So it should be either male or female. That's why any experiments here will be not really advantageous. And that's why this system is so conserved in this group. While in turtles, fish, and amphibians, this plasticity is much better observed. So about the strategies, let's skip this slide because it's something you already understand, that it's how the, because in some, just let me explain you something. Sometimes, like in the case of human, right, this is SRY gene on the Y chromosome, it expresses and it changes the fate of the embryo, so it determines the male pathway. While if there is a gene on W chromosome in some species, for example, in Xenopus here, this gene may be expressed and also it forms female. But for example, in birds, there are no genes on Y chromosome, on W chromosome, which affect sex. Actually, this chromosome is not much needed for sex determination. What is needed is the ratio of the Z chromosomes. And in this case, if there are two Z chromosomes, that the bird will become male. And if it's only one dose of Z chromosome, this bird will become female. The same happens in Drosophila. Two X's, female, just one X, male. And Y chromosome we can remove and we will not lose much, because the main thing is not the Y chromosome. The main thing is the ratio of the X chromosomes. So, about the genomics now and evolution, it's very interesting that in mammalian lineage, autosomal evolution is much faster than in marsupials and reptiles and fish. And if we want to see the most conserved part of the genome, is the X chromosome. When we compare the X chromosome, actually, look, this is the rabbit. This is, I think it's Camelus bactrianus. So it's camel, rabbit, it's pig, cat, um, how it's called, mink, and so on, elephant, human, they all are similar. Sometimes we cannot find similarity between autosomes of these species, but when we compare the X's, they are really similar. And it's so-called Onus law, that X chromosomes are really very conserved part of the genome. And this high conservation of Eutherian X we can explain by dosage compensation. Because of the inactivation of one of the X chromosomes in females, let's consider what if it will be a translocation, what we call chromosome evolution, if something is translocated on your chromosome. So if something is translocated on X chromosome, any translocation of chromosomal material will lead to the fact that this translocated material will be subjected to inactivation, which means if there are any dosage sensitive genes, that this part will be excluded. And besides, inactivated X has low late replication, so it will have effect on all early replicating genes. So that's why there should be a solution, and some additional mechanism must evolve that will prevent spreading of this inactivation somehow. And really, if we will consider different mammalian genomes, probably I hope you will work with some of indicated here mammals. And in some cases, we can see X autosomal translocation, and they will see that they survive perfectly well. For example, in some rodents, we see such translocation. We see them in some bats. We see them it's still unclear why, but we see these X autosomal translocations pretty often in artiodactyls, in cervids, in bovids, and in some insectivorants as well. But actually, any of these events, when we detect them, it's actually terminal phylogenetic branches, which means it's really recent events, which means maybe these are dead branches and they won't lead to anything novel. So let's consider now why autosomal translocations in Eutherian mammals. They are also pretty common in cetacea, but they also were described in carnivorous, in some primates, in New World primates. There are our relatives that carry Y autosomal translocations, and also in some rodents and edentats. Besides, as I already told you, in some rodents, the total loss of Y chromosome has also happened. And the main trigger of sex in therians is SRY gene. And what we must remember that this is the first discovered gene for, and the best described gene for vertebrate sex determination, because if we mutate this gene, it will lead to XY female. So these humans will look totally like female, but when we do karyotypic analysis, we see the Y chromosome, but the SRY gene on this Y chromosome won't work. And of course, these women will be sterile. If we inject SRY gene to mice, we will get XX males. So it will trigger the development of male pathway, but the chromosomes will be XX. And this gene is very old. We can find it in marsupials as well. Thus, we can calculate that it's appeared around 200 million years ago. And it's appeared from the gene SOX3, which is nowadays X chromosomal gene. So it's like appeared as one of alleles of this gene. And it's interesting that it hasn't been found in any other taxa except mammals. In some cases, there were found some version of SOX3 genes which also took this sex determination trigger role, but it's rather exception than the rule. So let's see here the human Y chromosome, which is also very well described. So mostly it's heterochromatin here. So with this part, we can delete without any big problem because we don't need it at all. So in some cases, in some human population, the Y chromosome is really, really tiny, but it's not a problem. So everything else is working perfectly well. It's just the heterochromatic block is missing. So don't be confused by that fact. And except for this trigger SRY gene, the Y chromosome also carries some genes which are important for male fertility. And they contain very large non-recombining part. And of course, the rates of Y chromosome evolution is very high. It's full of mutations. And we can observe genetic drift, decreased selection. And we see that for this 180 million years, it has lost almost all other genes which we'll now find on the X chromosome, except 200 genes which are located here on the very top of Y chromosome in the region we call pseudo-autosomal region. Okay, guys, I think I am a little bit late already. Do you still have ability to listen and understand me?
I don't know about another, but I have ability and I want.
Let's decide. I just have several slides because I will already round up and finish it soon. So let's continue, right?
Yes.
Yes.
Yeah, let's continue.
Okay. So let's see. Okay, we already were talking about sex-determined pathway. We won't talk about it anymore. So these are the species which I told you that rodents, they have really fast chromosomal evolution on all ways. Even autosomes are very rapidly developing. And this evolution is so fast that in some lineages, the Y chromosome has disappeared completely. Let's consider this genus Elobius. In Russian, we say Slipushonka. And there are 4 different species described. In one species, we can describe XX and XY system. So totally similar to most other mammals. But in Elobius lutescens, both sexes carries X0. How everything happens, how sex determination happens, nobody knows. So one of other genes has taken the role of sex trigger, and people trying to find out what kind of gene it is, but still many aspects are missing. Elobius talpinus, this species lives nearby Novosibirsk, for example. We've got many species from many individuals from this species. And all males we found, they all carried X and X chromosomes. And there is another species, Elobius trancrei, also very similar. So something strange happens in this lineage, don't you think so? And similar situation in another Ruicus spinneret, Takudaya, where they've got also X0 sex determination. So we don't find any Y chromosome, and in females, not 2 X's, but just 1 X. So how sex determination is triggered is really a tricky question. Probably it's on another autosome. So look what we found in our laboratory. We started to find lemon. Remember the colored lemon, those lemons which are, people say, suicidal and jump from cliffs, which is a legend, but a very nice one. Not very nice one. But nevertheless, it's a legend. But we are interested now in sex determination in this region. When we started to compare the autosome, we found homologous region between the autosomes and golden hamster and the field walt. So all autosomes were normal whatsoever. And we discovered also very fast evolution, but it's normal for muroid rodents. It's not something that should surprise us. What has surprised us really, it's this translocation. This is the X chromosome. This is male. This is the X chromosome. And this is translocation. And this translocation has another translocation here. And we call it X2. But this is an autosome as well. And where the SRY gene is totally missing here. And we don't know if this system is actually Y chromosome or it's just a translocated part. And this is still enigmatic, and nobody knows how the system works in Dicrostonyx starquatus, in collared lemon. And it also has many B chromosomes also for unknown reasons. In some cases, up to 24 B chromosomes were described in this species. And we suggested that Y chromosome is most probably lost completely and some new trigger has replaced SRY. And what was interesting, when we started to study the B chromosomes in this species, we found some X-specific genes. And maybe somehow these B chromosomes also play a role in sex determination, but it's only a big question. But in this case, anyway, B chromosomes have evolved from sex chromosomes. B chromosomes evolved from sex chromosomes, and something similar has happened in Drosophila in some cichlid species. In another species where we found the loss of Y is Lasiopodomus mandarinus. In this case, also we can find translocated X chromosomes and autosomes, and we cannot find any SRY gene or real Y chromosome. So all revealed cases of loss of Y chromosome are restricted to muroid rodents. And this is the taxon with really high mutation rates. So this is, we will just skip this slide. It's about acquisition and loss of mammalian escape of genes. It's kind of evolution, which we won't discuss today. I just wanted to tell you a little bit more about evolutionary parallelisms. For example, human, I told you that here we have SRY gene is homolog of SOX3 gene, but the same gene also, the allele of this gene also became sex trigger in one species of Parisia, in Madagascar gecko Pareodura, in true lizards of Lacertida, maybe, we don't know, but we suggest, and in some amphibians. Another case is sex chromosomal system in platypus. I already told you that it has 5X and 5Y chromosomes, and it's really interesting how they get segregated in meiosis, because all X's should go to one part, on Y's to another part. If we have 3 X's and 2 Y's going to one part, we will have a problem. That's why everything should be really precisely all X's to the left, all Y's to the right. And somehow the animal solves all these problems. So we studied the sex chromosomes of platypus and echidna, and what we have found that we didn't find any genes similar to X chromosomes, to eutherian, to human X chromosomes. All human X chromosomes is homologous to chromosome 6. So it means that totally different gene governs and triggers sex determination in platypus and echidna. And analysis of this group has helped us to reveal that actually this group is very old. This chain is more than 40 million years old. It's really very old chain. It means it's somehow advantageous. And some new elements were added in platypus, some new elements were added in echidna, but nevertheless, it's an old chain. And in this chain, DMRT1 gene is present because on the biggest chromosome, although it's not sex determining actually. Sex determining is AMH gene in platypus. But this DMRT1 gene triggers sex in such big group as birds, because it's like dosage of DMRT, which triggers the sex determination. Also in gecko hokuensis, in some turtles, in some frogs, and in Cynoglossus, in a bony fish Cynoglossus. Now MAH, which is now triggers sex determination in platypus, but it also triggers sex determination in some species of Atherina, in Nil tilapia, in Rana, in frog, and in Anolis sagray. We also found this gene. We don't know if it's triggered or not, at least it presents on the sex chromosome. So we will skip the triggers. We will skip how we analyzed one of Hecko species. Anyway, we found that this Hecko has this homolog of Anolis carolinensis chromosome 9, and the same homolog triggers sex development in iguanian genus Norops, in Pogonoviticips, in snakes, and in Xenopus levis. Okay, so let's skip snakes because we don't have time, and turtles. You can read it. I can give you articles if you'll be interested. Or you can just find it according to the to the presentation. And I just wanted to tell you very fast. Oh, okay. I just wanted to tell you very fast about the frogs, that sometimes in the same species, we can find several different sex chromosomal systems. Like in Rana rugosa, different populations might have XY or ZW system. And in such a way, since it's the same species, we can cross hybridize them, and we can get hybrids. And we can, for example, get XW or wild W individuals, and we can see which trigger is more strong. For example, if we have WY, if it will be male or female, and according to this, we see no, Y is stronger, it will be female. So it's a very interesting case, and that's why frogs now are very much a big topic of studying sex chromosomes, but they have mostly homomorphic sex chromosomes, but only genetic sex determination. Non-frog was found with environmental sex determination, which differentiates them from reptiles and many other groups. Okay, let's come to the conclusions. So our conclusions that Therian mammals possess very conserved sex chromosome system, but in some cases, maybe increased instability of X chromosome. And these evolutionary arrangements are still possible and are not fixed in evolution. Some species like arvicoline rodents exceptional because they can frequently lose Y chromosome and they might have unusual sex determination. And this case differentiates that very differentiated sex chromosomes may still escape evolutionary trap. So this trap is not the death trap, still can be escapable. Monotremes have an unusual system that have conserved sex chromosomal translocation chain, and some, yeah, I didn't tell you, but just believe me that some synthetic, this is the mistake, it's not systematic, synthetic blocks are frequently reused as sex chromosomes in different vertebrate taxa. And this should be taken into account when we compare different animals with unknown sex chromosomes. We already know what to search for. We know that only restricted amount of genes may become sex triggers. Okay, let's skip also further research. And these are the books I recommend you to read. Very funny books, you will read very easy. The Red Queen, very nicely written by Matt Ridley. I recommend you Steve Jones, The Descent of Man, also very nice book. Sex in the Sea, also very interesting book by Mara Hart. And Sex Chromosome, more scientific sex chromosome sex determination vertebrates, Solari, and I already told you about evolution of sex determination by Leo Boykboom. Okay, I'm ready to answer your questions. I'm sorry for this loads of information. I probably oversaturated your minds, but I really hope that all this.
Okay. No. I have a question.
Yes,
I have a question about. I forgot the name in English. Platanus, maybe этот. I mean.
Platypus.
I mean, utknos. Platypus. Yes. They have a five pair of sex chromosomes, and yes. Why? I understand that it's possible to get this in evolution, but why it is so stable? Because how I understand it is a lot of millions of years, it is so. There are 5 pairs, and why it's still so? Because I think that in evolution, possible when, for example, some XY and X7 could have a relocation, and during the evolution, it could be one chromosome because it's the less mistakes could be during the meiosis, but they still have 5 pairs, and why? It is a real interesting question for me.
Yes, you're totally right. Usually what we've got in evolution, for example, let's consider our X chromosome. We already know that our X chromosome is homologous to, I already shown you here, remember this picture, that our X chromosome has homology to chromosome of marsupial, but it's only Q arm. Look, the P arm was translocated after emergence of Eutherian lineage. So it means that our sex chromosome, our X chromosome, also was formed by a recent, which means like 120 million years ago, translocation of an additional element. So this formed our P arm, but the X arm of chromosome, it's still the same. So it means that X chromosome does change, but it doesn't form multiple sex chromosomes. Actually, multiple sex chromosomes are very rare. These are the only cases, like it's only less than 4, like maybe 10 cases for 6,000 species. It's very small amount. It means that really there is not a big advantage to have multiple sex chromosome. And we should expect that soon after this chain appears, it might somehow get resolved in evolution and so on. But it's not the case. We really don't know why and how monotremes have retained this ancestral chain. Probably they evolved something specific, and probably somehow this specific thing which they evolved, it put them into evolutionary trap. When we see that something is not changing for a very long time, it means that this changing may be not very good. Maybe they bring into something detrimental. That is,
yeah. I heard one important. If something is not changing during the evolution, you should ask the waterfare what's the function. So this because it's important in evolution. Yeah, thank you for your answering.
You're welcome. Okay, guys, if there are no more questions, then I think we can slowly finish our seminar. I thank you all for your attention, and I hope to see you again. Maybe we can still talk about gene amplification and segmental duplication. We can have just a short seminar. Please talk to Alexei. Maybe we can organize it somehow just to finalize our discussion about genome redundancy.
It would be very nice.
Okay.
Okay.
So
The lecture was really cool.
okay, thank you.
Thank you for your lectures.
Thank you for lectures. Yes. And
welcome.
See you.
For questions and for your attention. And I hope to see you soon again. And have a nice day. And
you too. Yes. A nice night. Maybe. Goodbye.
Goodbye.
Bye.